Hi everyone, and welcome to your first video for Year 12 Biology. This one's going to be on Chapter 2A and 2B and looks at the relationship between nucleic acids and proteins. So I'm hoping that you guys picked biology because you're already interested in it. You might have done Year 11, and hopefully you know that biology is all about the study of life. And it's really difficult to understand how life works without understanding proteins. Proteins are like the building blocks of life, and they carry out so many different functions in our bodies, like digestion, building muscles, immunity, and many of these processes we're actually going to look at throughout this course. So it's going to be really important that you understand what proteins are and what they can do. It's also important to understand how they're made, and to understand how they're made, we need to also know about DNA. So DNA holds all the instructions for all the proteins that are eventually going to go on and carry out all these different functions. You can sort of think of DNA as like the blueprints or instructions, and the proteins as the machines that are created that are going to carry out all of those functions. So let's find out what these molecules actually are and what they do. So the first study design dot point is about understanding proteins and understanding what the proteome is. So the proteome is just the collection of all proteins within an organism. So you might have heard of the genome, which is all of the genes in an organism. The proteome is just the protein equivalent. So proteins are macromolecules. So you might have heard of these before. We're going to focus on two in Year 12 biology. We're going to focus on proteins and nucleic acids. But you might also remember that there is lipids and carbohydrates as well. So they are a macromolecule consisting of amino acids. So amino acids are the monomer of proteins. So the monomer basically means just the subunits or the building blocks for the proteins or for the polymer. So amino acids act as the building blocks for the building blocks, which is nice. And they're linked together by peptide bonds, which we'll learn about in a second. But the takeaway from this slide is just understanding a couple of the key roles that proteins can play within our bodies. So we've split them into 7 different categories. So they can be immunological. So they can help the immune system to defend the body against diseases. They can be contractile, like we talked about, they can help with building muscles and contracting muscles, things like that. They can be involved in storage of particular molecules and chemicals. They can be structural, so for instance, collagen can provide structural support to our tissues. They can act as a transport system, like hemoglobin, which transports oxygen. They can be catalytic, which we're going to learn about in a bit more detail in Chapter 3, but they can basically act as enzymes and facilitate reactions. And they can be signaling molecules, so things like hormones. They can act as communication systems from one cell to another. So now that we know a little bit more about what proteins are, we're going to dive into what they look like and what their structure is that helps them to be a functional protein. So as we talked about before, amino acids are the basic subunit from which proteins are made. Okay, so we have many different types of amino acids, which we are going to look at a little bit later on. But they form this, this was called a primary structure. Okay, and this primary structure or the sequence of the amino acids here is what is controlled by DNA and genes. Okay, so what we read on the DNA is what is going to code for this amino acid structure that forms here. And as we read before, these are all linked by something called peptide bonds. All right, so you've probably heard of amino acids before if you've looked at things like if you're into like nutrition, or if you go to gym or whatever, they might talk about, getting amino acids in your diet will help this, and this, which is, not wrong. Amino acids are something that we do have to require from our diet. So plants make their own, but we have to, eat certain foods, and that's why eating proteins is so important to get the essential amino acids that we need to be able to create all of these functional proteins that we need to survive. So that's why amino acids are so important. Now, their structure. So there's 20 different kinds. Okay, so there's 20 different types of amino acids that are found in all proteins in all living things. So this is the common structure on the right-hand side here of any generic amino acid. All right, so this bottom bit here, okay, so this nitrogen, this hydrogen, and this carbon dioxide group here, and this central carbon, So this is all very consistent amongst all amino acids, okay? They all have an amine group, they all have a hydrogen, a carboxyl group, and a central carbon. What makes each of the 20 different amino acids different is this R group here, okay? So this R group is what varies chemically from amino acid to amino acid to make them unique. And we're going to go through some of those different um, types of R groups now. So you can see here we've got 3 different amino acids. Okay, you don't need to know the names of any amino acids. They're just 3 different types. And you'll see that they've all got a different R group. Okay, everything else is the same and consistent, but they all have a different R group. And this is what makes them all unique and all different amino acids. So for instance, this first one, it can form disulfide bridges with different other amino acids. This one has alkaline properties. This one has acidic properties, which means that they're going to do different things and they're going to fold differently and attract different things as well. So this is what makes a protein shape and function different, and that's why the R group is so important, because it brings that uniqueness to the function, and we can have many different proteins that do many different things in our bodies. So the structure of the whole protein, or sometimes called the conformation, okay, so conformation, if you see this word, it just means the shape or the structure of something, is dependent on a variety of things. So there are 4 levels of protein structure that we're going to learn about, okay? So we've had a quick look at this one. We're going to dive into it more deeply, and then to the fully functional protein at the end. So we're going to look at the 4 levels of protein structure that require to carry out its biological role. So the first hierarchical level that we're going to look at is the primary structure. And we looked at this a little bit when we're talking about amino acids, because basically what it is just the sequence of amino acids that is coded for by DNA. Okay, so it's its simplest form. It's just a bunch of amino acids linked through peptide bonds, all right, which are formed via condensation reaction, and it is the simplest structure a protein can be. So some proteins can actually just be this. They don't go through any other folding or anything like that. But this order, okay, the order of these amino acids is really, really important for how it's going to function. And later on, when we look at mutations and things like that, you'll see how this order changing can severely impact a protein. All right, so the primary structure is just the amino acid sequence that is joined by peptide bonds. We then go into our secondary structure. Okay, so the next level up, if you will, is the secondary structure. So there's 2 different types of secondary structures that can form. We can have alpha helices, which is these spirally things here, and we can have beta pleated sheets. All right, so it all depends on our carboxyl and our nitrogen group. So do you remember that consistent portion of the amino acid that I showed you before? This group here and this group here is what's going to determine whether or how these shapes form. So it's interactions between those 2 that are going to form the helixes or that are going to form the sheets. So this is the secondary structure. So our primary structure can go onto here, but they are linked by hydrogen bonds. All right, so that's probably the most important thing to take away from this, is that our primary structure, through these interactions with these 2 groups and through the formation of hydrogen bonds, can form our secondary structures. Then from there, these secondary structures can go on further into our tertiary structures. So this is further folding and interactions between different structures that form the folding of this tertiary structure. So this is where our R groups come in. So remember that these super unique R groups with all of these different properties like alkaline, acidic, hydrophobic, whatever, they form all of these different kinds of folding. Okay, so you can see that the tertiary structure is made up of several different things. Okay, so you've got your alpha helices or your beta pleated sheets are going to be intertwined in there, and then depending on the R groups in those amino acids in that primary structure, you're going to have bending, folding, shaping, formation of bonds. There could be disulfide bridges, there could be hydrophobic reactions, there could be a bunch of things going on here based off of the R group that has got to cause that folding into the tertiary structure. Now, many proteins, this is their final form, okay, so this is as far as they go, but for some proteins, they need the quaternary structure. So this is the fourth level, or as far as a protein can go. So this is when a protein needs several tertiary structures joined together to form its final functional shape. Okay, so you can see here the example of hemoglobin. Hemoglobin is made up of 4 different separate tertiary structures that bind into one big functional unit. Okay, so quaternary structure is just the joining of other polypeptide structures together to form the final protein. Okay, so that is proteins. Okay, so that's what they do, and that is the structures that they can take on. So now what we're going to look at is we're going to look at the instructions or the blueprints that are going to help determine those primary structures and the formation of this, the functional protein. So we're going to be looking at nucleic acids and DNA as the instructions for proteins. So hopefully a diagram on the left-hand side here looks a little bit familiar from year 11. So it's just showing you when I'm talking about DNA and genes, this is what I'm talking about. So you would have learned at the cellular level, the nucleus holds the DNA in the formation of chromosomes. And if you unwind this, you find our DNA in these helical segments, and then within those are our genes. Okay, so we're going to be diving even deeper than that, and we're going to be looking at the individual little ladder parts of the DNA. Okay, and they're what are called the nucleotides. So just like our proteins had amino acids as their monomers, our nucleotides and DNA also have a monomer, which is called the nucleotide. Okay, so these are the building blocks of DNA. So below here is a generic diagram, just like the amino acids, of a nucleotide and the 3 main components that are required. So you will see that all nucleotides have a phosphate, okay, which is going to be really important when we start to actually join these together. So everything has a phosphate group. All DNA has a sugar called deoxyribose, okay, and that's where the D in DNA comes from. If it's RNA, which we're going to learn about as well, it'll be a ribose sugar. All right, so already that's a key difference between DNA and RNA, but all nucleotides have a sugar of some description, and all nucleotides will have a nitrogenous base. So depending on what nucleotide it is and whether it is RNA or DNA, the base is going to be different. So there is 5 potential bases that it can be, and we'll talk about what those are and when they're going to come up a bit later on. But this is the generic nucleotide structure, and you will need to know or be able to recreate this generic structure with the phosphate, the sugar, and the nitrogenous base. So how do these nucleotides form? All right, so they aren't just hooked together by magic. There's obviously a bunch of chemistry and bonds and things happening around this. So nucleotide is formed when a chemical bond between the phosphate molecule and the sugar molecule forms, and this is called a phosphodiester bond. Okay, so these are the bonds that create that ladder structure. So when we look at DNA, and this is a really bad drawing, this ladder structure that we see, okay, this backbone here, that is formed and connected by phosphodiester bonds. Okay, so that's how these are chemically bonded together, which is going to be important when we look at how DNA unwinds and breaks apart a little bit later on. So we talked about these nitrogenous or nucleotide bases, and there was 5 different types. So there are 3 types that are consistent between DNA and RNA, and then you'll see I've highlighted in green a base that differs between DNA and RNA. Okay, so DNA is our double-stranded genetic information, and it consists of 4 bases: adenine, guanine, cytosine, and thymine. RNA is our single-stranded basically replica of DNA. So RNA is what we use because we obviously can't use DNA. DNA can't leave the nucleus. So if we are wanting some instructions to leave the nucleus to go out and build proteins and things like that, we form single-stranded RNA. So RNA also has adenine, guanine, and cytosine, But you'll see in RNA that thymine has been swapped with uracil. Okay, so that's another key difference between DNA and RNA. So on the right-hand side here, you can see that they're split up into two different groups. We've got our purines and our pyrimidines. Okay, so we've got adenine and guanine as our purines, and we've got cytosine, thymine, or uracil, depending on whether it's DNA or RNA, form our pyrimidines. So a really important thing to note is that a purine will always pair with a pyrimidine, and a pyrimidine will always pair with a purine. So these are the pairings that will always occur in DNA. So cytosine will always pair with guanine. Okay, that there's no exceptions to that rule. Cytosine will always pair with guanine. Adenine will always pair with thymine. Okay, no exceptions to the rule. Adenine, thymine always pair together. RNA, of course, we don't have thymine, so that thymine is changed to uracil. Okay, so RNA is the same for guanine and cytosine. They always pair together. But then in RNA, it's adenine and uracil that pair together. Okay, so it's still the same idea, purine with a pyrimidine. We're just swapping that thymine for uracil. So you can see here what I mean, okay? So here I've got a DNA structure, a full DNA structure, okay? We've got our phosphate, we've got our sugar, which is a deoxyribose because this is DNA, and then we've got our bases. So you see here that G is paired with C, T is paired with A, okay? If this was RNA, we would change this for a U, but this is DNA, so we're going to leave the T as is. But that is the pairing. So you just really need to remember that those are the pairings that DNA has no matter what. Okay. You'll also see a couple of things to note here is that you can see that these strands are running anti-parallel to each other. So you can see that this is going in this direction and this is going in this direction. Okay. So they're kind of like back to front, running either side of each other. That's what's formed the lattice structure of our DNA. You can also see these phosphodiester bonds here, like I talked about before, that is linking what we call the sugar phosphate backbone. Okay, so that's what's linking all of these together and keeping that lattice structure intact. You can also see that our bases are connected with hydrogen bonds. Okay, so hydrogen bonds is what keeps our nitrogenous bases together and forms that beautiful helix structure that we looked at. That's going to come in handy when we look at breaking those apart later on in our course, but they are formed through hydrogen bonds, our GC and our TA pairing. And this is what forms our complete DNA structure. So when we see that DNA ladder structure and we see all those little moving parts in there, this is just a zoom in on one of those parts of the ladder. Okay, so we've got our sugar phosphate backbones, we've got our bases that are connected with hydrogen bonds. That's the basic generic DNA structure that you'll need to be able to replicate. So like I said, all right, we've got this sugar phosphate backbone here, and each individual nucleotide needs to be able to connect to the next one. Okay, so how do these connect? We call this a dinucleotide. Okay, so di just means two, so it just means two nucleotides attached together. If we were to keep going with this, we'd have another one. I can't draw very good, so that's another one. Then we'd have another one, etc., to form that DNA ladder structure. So dinucleotides are formed when two nucleotides are covalently linked together through a condensation reaction. Okay, so condensation reaction just means a reaction that releases water, and this is how we link those phosphodiester bonds together to form that sugar phosphate bond. So now let's have a look at RNA structure. So you'll notice that RNA structure is obviously very, very similar, but there is a couple of key things to note that are different, okay? So obviously the first thing that is different is, where's my pen, is we have a different sugar, okay? So RNA or ribonucleic acid is a single-stranded version of the DNA. So remember I said that DNA is the instructions. It can't leave the nucleus. So we've got this single-stranded RNA that is created to leave the nucleus and take the instructions elsewhere. So the first difference is that it is single-stranded. Another difference is that we have the ribose sugar here instead of the deoxyribose sugar. So it's a different sugar that we use in RNA. You can also see that we have replaced our thiamine with uracil. Okay, so that's another key difference that we've learned about. And like I said, it is single-stranded. Okay, so that's another key difference. So like it says here, RNA has many functions. When we learn about how proteins are actually made, this is when RNA comes into play. And there's 3 different types of RNA that we might be asked about. Okay, we've got our messenger RNA, which we're going to look at a lot in our next lesson. So these are transcribed from DNA and acts as instructions or mobile instructions that can move around and carry instructions outside the nucleus to make proteins. We've got transfer RNA, which we're also going to learn a lot about in our next lesson, which is a type of RNA that will deliver amino acids to create that primary structure that we learned about before. And then we've got ribosomal RNA. Okay, so this is something that is specifically attached or inside of ribosomes, which again we're going to learn about very shortly in our next lesson. So this is just a little summary slide because VCAR often loves to ask you about the similarities and differences between DNA and RNA. It's one of their favorite questions. So I thought we'd summarize in a nice little table for you. So we've got our DNA here, we've got our RNA here, and we've got all of these different categories in which they differ. Okay, so like we said, double-stranded versus single-stranded. They have a differing sugar. They have that one differing base, the thiamine to uracil swap. And DNA is our long-term storage of instructions, whereas RNA is a temporary short-lived copy of those instructions. Okay, so if you have any questions, bring them to class and I will see you there.